Okay, I think I'll get started with the introductions. I'm so happy to be hosting this meeting of the LRD. And today we're going to be talking about a phenomenon that's really central to my research and of great interest to me. Historically thinking, it's interesting to me that in the 1960s, at the same time when the function of brown adipocytes was being identified, and people were starting to realize that they had uncoupling and starting to get hints of uncoupling protein 1. At the same time, there was just a plentiful plethora of research on this very interesting phenomenon of triglyceride cycling that occurs in adipocytes and accelerates when lipolysis is activated. And I think that this is something that we still have a lot that we don't understand about the physiological importance of this process of triglyceride cycling and what it does to the adipocyte and what the consequences are systemically. So that's why I organized this meeting based on this topic, and I hope you can appreciate you're going to get kind of two different viewpoints of this cycling from Anand and I today. So Anand Sharma is a fantastic scientist. He did his postdoctoral work with Christian Wolfram in Zurich, and he's currently a senior scientist there. But just at the beginning of next year, he's going to be moving to Imperial College London and starting as an assistant professor. So congratulations on that momentous move forward in your career. And please tell us about what you found out about triglyceride cycling in adipocytes.
Thanks a lot, Shannon, for this lovely introduction, warm wishes, and also the invitation in the first place for me to present on this amazing platform. And thanks a lot for joining, everyone. And I think this audience will resonate with me that adipose tissue is a remarkable organ. And for a long time, it's sad that people always thought that adipose tissue is kind of inert energy storage tissue, that's it. But now we know it's not an inert organ. And also it's not just a lump of mass, rather where the adipose tissue is that affects how we feel metabolically. And that's why the concept of metabolically healthy and unhealthy obesity arises. But in the more recent years or let's say more recent decades, one thing which has been, again and again recognized is that the dynamicity or dynamics of adipose tissue may be a crucial component of metabolic health. So it's not just how much of adipose tissue we have or where we have, rather how dynamic the adipose tissue is. And the bidynamicity, what I mean is exactly what Shannon just introduced, the cycling of adipose tissue. So in general, when we are fed in the fed state, we have to store the excess energy in the form of fat. And after some time, with our body, muscles, and other organs, they need energy. And that's why the stored fat has to be hydrolyzed into smaller units, preferably fatty acid and glycerol, and it's supplied to the tissue. But what is more surprising is that maybe like 70 to 80% of these hydrolyzed fatty acids, they are re-esterified back into the storage form, which is triglyceride in our body. Now the question is, this re-esterification of fatty acid is very energetically expensive process. To recycle one triglyceride molecule, we need at least 4 ATP molecule to be invested. Now, if we are hydrolyzing and re-esterifying 80% of total fat back, why would evolution favor such futility? It has to have some function in the tissue. So that's what we are looking at. That's one part. And second part is when we look at adipose tissue, we generally categorize them into two types. One is white adipose tissue, and other one is brown adipose tissue. The question is if we talk about futile lipid cycling, do they both cycle at the same rate in the same way, or are there some differences? And if there are some differences, what are those? And in fact, if we look at the adipose tissue morphology, I think it's a very interesting feature of metabolic health. We have white adipose tissue where the adipocytes, they show one big unilocular lipid droplet in the cells, while the brown adipocytes, they have multiple smaller lipid droplets in each cell. And morphologically, this is one of the best defining feature of brown and white adipose tissue, the morphology. But if we go deeper, we see that the UCP1 expression is more prominent in these multilocular adipocytes. And probably most of the researchers, we stop here that when we say this is brown adipose tissue or beige adipose tissue, we just show the morphology and we show UCP1 expression. But some people go ahead and show that the sympathetic innervation and mitochondrial density is enriched in the brown adipose tissue, and that's one of the defining mark. But it doesn't seem to be that simple in physiological scenario, because if we expand our experimental space, we see that this kind of simplistic characterization doesn't hold true for all systems. For instance, this is mouse adipose tissue. So we see epididymal or inguinal adipose tissue, which are white adipose tissue. They have unilocular big lipid droplets, while brown ones, as expected, they have multilocular lipid droplets, adipocytes. What is fascinating is when we look at the UCP1 knockout mice, we look at the inguinal white adipose tissue, and if I did not tell you that these are UCP1 knockout mouse, you would, or I would immediately say this is classical Beijing phenotype. But if we define Beijing based on UCP1, I would say it's wrong to call it a Beijing, because these tissue do not have UCP1. On the other hand, when we look at the brown adipose tissue, some of the cells, they become unilocular like white adipose tissue. So this tells us that actually beyond UCP1, there is something else which kind of defines the adipose tissue function, whether we call it brown or beige. And the question is, what defines this catabolic nature? Because in the UCP1 knockout mice, for example, we know that the inguinal adipose tissue is highly catabolic, more than the brown adipose tissue in UCP1 knockout. So I think the functional definition of catabolic, the beige adipocyte should be on the catabolic activity of these cells. And that brings us to the question, what are the mechanisms that make this white adipose tissue so catabolic? And is it actually dependent on UCP1 or not? And clearly it is not. And in the last couple of years, and especially in last one decade, especially Leslie Kozak's lab and other labs, they have shown that there are multiple mechanisms that are activated in these IVAT tissue, and that's how they become catabolic. And one of the mechanism which is frequently observed is futile cycles. Futile cycle of calcium, futile cycling of creatine, and sometimes futile cycling of lipid. And the lipid futile cycling will be the topic of interest. And although I know most of us, we already know, for the new audience, I would like to quickly go through this classical definition of what what a futile cycle is. So any reaction where two opposing enzymes, they convert one substrate to product and back to substrate, wasting one ATP molecule, that will be called as a futile cycle, because we are wasting ATP molecule. And this can go on perpetually, and that's why it's called cycle. So we waste cycle recurrently, and that's why it's called futile cycle. One classic example could be phosphorylation of glucose by glucokinase or hexokinase using ATP molecule. And if we have this phosphatase present, it will just remove the phosphate group. We start with glucose, we end up with glucose, producing nothing, classical futile cycle. And these futile cycles can be as simple as just flow of ions from, let's say, calcium flow from Surka channel, or it can be as complex as the central dogma of molecular biology. So the whole RNA synthesis, protein modification, protein folding, everything can have a classical futile steps. So almost every pathway in our cell has a potential to run futile cycle. We focus mainly on triglyceride or lipid futile cycle because this is one of the most energetically expensive futile cycle. In recycling one triglyceride molecule, we waste um, at least four direct ATP molecules because to activate each fatty acid, we need one ATP molecule. So while studying futile cycle in Christian's lab, we focused almost every prominent step of the lipid metabolism pathway. Of course, there are some interesting observations, but what at the end we noted, and probably Shannon will show you more from other side of the cycle, what we found that the triglyceride to diacylglycerol cycle is one of the most pronounced step in the futile lipid cycling pathway. And that's why, although we have generated a lot of data from many other steps, I would focus more mainly on diacyl to triacylglycerol pathway. So the lipolytic stress step or the hydrolysis step is catalyzed by ADGL to generate free fatty acid and diacylglycerol molecule, while DGAT1 and 2, they use fatty acyl-CoA to resynthesize triacylglycerol and wasting ATP molecule in a futile way. And this was quite challenging question because until very recently, when in 2023, Christoph Thiele's lab published this methodology, we did not have a very good method to actually quantify the rate of lipid cycling. So we collaborated with Christoph Thiele, who developed this method. And in the very simplistic term, what we do is we feed the cells with these three fatty acid, tracer fatty acid, which are non-natural because they have this terminal alkyne bond, and our body or natural lipid fatty acids, they do not have this bond. And this is used to distinguish these tracers from endogenous fats. And when we feed these fatty acid into the cells will be synthesizing new triacylglycerol molecule, and some of them will have all the three labeled tracers, some of them will have two, and some of them will have one. And this will be, we can plot it at the time zeros, the cell acquired, let's say, 55% of single labeled species, and then 40% of double labeled, and then 5% of triple labeled species. But if there is futile cycling going on, this triglyceride molecule will be hydrolyzed, and this would lead to reshuffling of these tracer fatty acids. So what we will see is over time, we see increase in these single labeled species, and in parallel, we see a decrease in these dual and triple labeled species. And this is the most direct and quantitative method to measure futile lipid cycling. And then we can use these data and deconvolute it to derive other parameters, absolute parameter, for example, the T half of any given tracer fatty acid. We use this methodology, and I would again focus on this triacyl to diacyl step. When we block this ATGL step, we see in the control cell this appearance of monoacyl single label species is this much. But the moment we block this ATGL step, this line is completely flat. That means the cycling is mostly gone. So it's not just the presence of fatty acid from the external source, rather the hydrolysis of endogenous fatty acid or triacylglycerol is probably also equally required. When we look at the opposite step, which is synthesis of triacyl from diacylglycerol using DGAT1 and 2, we see the completely opposite of what we should see. So in this graph, this is single label appearance of single labeled species. We see blue line over time. This is control. The appearance of single labeled species goes up. The moment we add isoproterenol, which is inducer of lipolysis, we see this massive increase in the appearance of single labeled species. And the same we can see the disappearance of triple labeled species. So it's very consistent. The moment we add this inhibitor of DGAT1 and 2 combined, in the basal state, we blunt, to some extent, we blunt the appearance of single labeled species. But when we compare this isostimulated stage, there is a very pronounced reduction in the rate of cycling compared to the isostimulated stage. And this brings to, almost back to the control level. So almost all of the cycling is completely blunted in this system. And this suggests that actually the ATGL step as well as DGAT steps are equally important to run this futile cycle in adipocytes. Another feature of this methodology is we can measure the modification of these fatty acids because we can distinguish these fatty acids from the endogenous ones. We can see how much of the modification, which it can include elongation or unsaturation of fatty acid, happens over time. And again, we see the blue line, which is control. The moment we add DGAT inhibitor, it kind of suppresses the modification, and which suggests that one of the function of futile lipid cycling is actually to facilitate elongation and unsaturation of the fatty acid. Once again, we see that when we have this isostimulated stage, that kind of encourages more and more fatty acid modification. But when we block DGAT step, this kind of completely blunts it. So with this, what we found that DGAT 1 and 2 are key restrifying enzymes in adipocytes, while ATGL is kind of key hydrolase in this system. And this is not just for one cell line, it's true for many other systems. So we used brown adipocyte cell line, we use 3T3L1, we used primary brown white adipose tissue as well as human derived immortalized brown and white cell lines. And in all of these systems, if you just focus on PC1, which is the half rate of futile lipid cycling, what we see that compared to control, which is in blue here, the moment we add isoproterenol or CAMP in case of HMADs, we see that the curve shift towards left, which is shorter cycling rate. That means that these cells, they start cycling at faster rate. And the size of these symbols corresponds to each cell line. So you see most of the cell lines, they behave in the same trend. And the same happens when we block ATGL, we see the curve shifting towards right, which means they are slowing down the cycling, and the same almost similar happens with the DGAT inhibitors, although we see some cell type differences in these with these DGAT inhibitors. So with these, what we found that actually the adipocytes, they kind of decide which fatty acid is close to endoplasmic reticulum, and only the adipocytes, the lipid droplets that are close to endoplasmic reticulum, they can access DGAT1 preferentially, and that's the fatty acid that's mostly destined for restrification. So this was all cool. But what was not clear that the thing that we started with, that we have brown and we have white adipose tissue. While white adipocytes, they have one big lipid droplet. Brown ones, they have multiple smaller as well as larger lipid droplets. The question is, do all these lipid droplets, big and small, do they all cycle at the same rate, or there's some difference in their cycling propensity, or some mechanistic difference in cycling? And this is very crucial because we know if we just look at the sphere and if we assume that lipid droplets are spherical, the larger spheres have lower surface to volume ratio than the smaller smaller spheres. That means the smaller lipid droplets will have more surface area, which can be accessed by enzymes, and probably they can, cycle at faster rate. And to test this hypothesis, what we did, or let's say Christoph Thiele, who's the leading collaborator in this project, what he did is he made two scenarios. He assumed that all the cells in the system, either they have one single lipid droplet pool, all of them they behave similarly, or he modeled that there are two different pools, one cycling at faster rate and other cycling at slower rate. When we look at the fit here, we see that compared to one pool model, we see that the data, actual real data, it fits better in the two pool model. And this suggested that probably in the adipocytes, especially the cultured adipocytes, we have two distinct pool of lipid droplets. One probably that cycles faster, while other one that cycles lower. And in the graph on the right, each dot is actually just the least square some value of this fit. And any value which is higher than 1 suggests that two pool model is, explains the data bit better than one pool model. And if we see that in all the cell lines, each dot is one experiment, and we see that almost every time, we see most of the time, all the adipocyte models, they show two lipid droplet behavior. Only some of the 3T3L1 cells here, they show lower value than 1. That means that these cells, 3T3L1s, they have one homogeneous lipid droplet pool that all cycle at the same rate. And we know that 3T3L1s, they have generally, homogeneous big lipid droplets. So this data kind of explains very well what we observed and hypothesized. So to explain this difference in lipid droplet cycling rate, what we thought that these mature lipid droplets, which are bigger in size, probably they cycle at the slower rate because they have lower surface area compared to the small lipid droplets, which are still on the ER, or just say, nascent lipid droplets coming out of the ER. So they will have all the enzymes enriched on the surface area. And to test this hypothesis, what we did is we first, just simple experiment, we labeled the fatty acid or tracer lipid droplets with the labeled fatty acid. And what we see that the first fatty in the early time points, the fatty acid shows perinuclear diffused staining, which corresponds to endoplasmic reticulum. While at the later time points, we see this tracer enriching in the lipid droplets. So it's kind of indirectly suggests that probably the new coming lipid droplets, they are the ones that first acquire this tracer, and the reesterification probably doesn't happen a lot on the mature lipid droplet surface. And just to revalidate that this model is true, we kind of fractionated the mature lipid droplet fraction versus the membrane fraction, which has this endoplasmic reticulum and small lipid droplets. And compared to mature lipid droplet, you see this blue line, the membrane fraction cycles at way faster rate than these mature lipid droplets. So it's kind of proves that these small lipid droplets, which are still close to endoplasmic reticulum, they cycle faster than the mature lipid droplets. And if you are wondering how fast and how slow we are talking about, here is a true data from HMAT's human adipocyte model. So in the control condition, the first pool, which is faster cycling pool here, it takes 2 hours to half cycle it, while the other pool takes 8 hours. The moment we add CAMP, this pool is 4 times cycling 4 times faster, so it takes just 30 minutes to cycle half of it, while the 8 hour becomes 2 hours here. So both the pools, so what this data suggests that after adrenergic stimulation, both the pools of lipid droplets, big and small ones, they start cycling faster. But the difference between these two pools, 2 versus 8, which is fourfold difference, this difference is kind of maintained even in the stimulated state. So yeah, this is all very cool data. We have done this elaborate study in the mice. We generated DGAT1, DGAT2 single with UCP1 double knockout, as well as UCP1, DGAT1, DGAT2 triple knockout. And this study also, which is almost accepted in cell metabolism, hopefully, this data also revalidated that futile lipid cycling does have a physiological function, at least in the mouse model. And yeah, these data are very exciting. We think that these lipid droplet heterogeneity is one of the key features that defines the tissue specific differences. But we should not forget that it's not just the lipid droplet diversity. The diversity in adipocyte itself can have a huge influence. For example, in this study led by Tong Tong, and I was part of it, what we found is when we keep mouse in UCP1 knockout mouse in cold, we see that compared to room temperature, we see this P2 subpopulation of adipocytes emerging after cold exposure in UCP1 knockout. And this UCP, this P2 subpopulation is enriched in futile lipid cycling as well as other futile cyclic pathways. So this kind of P2 adipocyte seems to be, specialized adipocyte subtype that uses futile lipid cycling and other futile cycles. And it's not just in mouse. When we look at the human data, we found that actually there is this corresponding subpopulation called HAD3 in humans, which resembles this kind of P2 subpopulation. And again, in human as well, this HAD3 is positively linked to energy expenditure. So it's not just the lipid droplet heterogeneity, rather the heterogeneity of adipocytes as well. And this is not just one-off study. Another paper from Spiegelman group showed that there are some specialized cells that are enriched in futile creatine cycle. And more recently, in this Nature paper from Imperial College London, Will Scott's group, they showed that there is, after weight loss in obese individuals, a new subpopulation of adipocytes emerges, which are enriched in this reesterification and lipolysis machinery, which is the elongated way of saying that these are again enriched in futile lipid cycling. So with this, I would like to conclude that in when we talk about adipose tissue is not, of course we all accept that it's not just inert organ, it's very dynamic. But when we talk about details of dynamics, the diversity of adipocytes and the diversity or heterogeneity of lipid droplets within adipocytes is a key factor that we should not forget. And most likely the depot differences, especially in the context of knockout mouse, they may be arising because now we have multimolecular lipid droplets that can cycle faster. And probably this is one of the way by which UCP1 knockout mice, they compensate their thermogenic need. So with this, I would like to thank Christian Wolfram, of course, for hosting me and, amazing support, amazing mentor all these years. Radhika Khandelwal, who's also my partner in life, in personal life, and also a collaborator in science for last 10 years. So thanks, Radhika. Yelena from Christoph Thiele's group. These two people, of course, most of this project would not be, a reality unless we had collaborated with these two amazing scientists. Of course, all the Wolfram Group lab members, SLA people, and thanks all for your attention and time.
Thank you, Anand. That was excellent. I'd like to open it up to questions. If people would like to ask their questions, you can raise your hand and I will give you the ability to actually ask your question out loud. So. Okay, now.
Maybe I will stop sharing them.
Yeah, sorry, I'm Kalsum, can you help me? I'm having a hard time talking for a minute. Thank you. Daniel, will you please ask your question?
Sure, that was a great seminar. I really enjoyed it. I wonder if I don't remember if you know of any data that suggests that the DGATs or even UCP have a dependence on curvature, and that may reflect some of the data you're saying that the curvatures of the smaller structures are going to be higher than the ones in the larger one. Do you know if you know what I'm talking about? Is there any data that curvature plays a role in this?
To be honest, in one line, no. We were thinking mostly, or I was thinking mostly from the surface area point of view, but that's a great point, and I will think about it, the curvature, and also if that kind of affects the targeting of DGAT, especially DGAT1 to lipid droplets. So I will think about it and consider this possibility.
Yeah.
But yeah, maybe I wanted to add something more that when the cycling runs, we see unsaturation and elongation of fatty acid. And that kind of, if that goes to LPC pool or even in the mitochondria, these are very essential for, you know, higher curvature membranes, so including mitochondrial cristae or smaller lipid droplets. So I think it could be a feedback as well to make more of unsaturated and elongated fatty acid that in a way facilitate more curvature.
Yeah, I think that's a good point, but that I think the structure of DGATs, I could be wrong about this, I may be misremembering this as well. I think that in the structure of some DGATs, or at least DGAT1, the active site is actually a curved kind of interface. So the curvature may play a role.
That's a great point, and I will definitely look it up. And I mean, it would be consistent, to be honest, if that works that way. But it's
Right.
a great point. Thanks.
Right. Very good.
I think we have one more question. You can go ahead, Dr. Yang.
Hey, Shannon. Yes, good to see you. I remember we worked together at the ASBMB. Yes, this is a great talk. I have a question about the two-phase lipolysis. With in relation to large and small lipid droplets. So in real adipocytes, right? So there's only one unilocular lipid droplet, and my question would be that if you could do your analysis in such a the real adipocyte where there's one giant unilocular LDs and perhaps many small little LDs, then the rate of difference in lipolysis would be much bigger, right? I mean, like now it seems to me that you use cultured cells where you have it's a population of, I mean, the size of the LDs are kind of heterogeneous. I mean, the size differences are not as big as, I suppose, the real adipocytes.
Yeah, that's a good point. I mean, I completely agree, and we are right now. We are maybe, let's say, halfway through to optimize the protocol, at least to do it in mice, live mice. Of course, it takes way more of these tracer fatty acid, and we are injecting this triple label, which Christoph synthesized. So none of these resources are available in market, and it's a tedious process to synthesize these tracers. But hopefully we are close to optimizing the protocol in vivo, at least in mouse. Humans, I think that's still a long shot, because the moment we isolate SVF is not true adipocyte anymore, while culturing adipocyte will be a bit tricky. But mouse data definitely soon we will have. Yeah, it's good point, and I think soon we will have the true data to answer this question. Thanks.
All right, thank you.
Yeah, thank you for those questions. Those are really interesting. I would just build off of that last question. If you had a single lipid droplet, what your model is really saying is that there's different pools of triglycerides that are cycling at different rates. And it's not inconceivable that you might have different locations on that lipid droplet surface that may behave differentially.
Yeah, I think so. Especially in one of the reviews in Nature Metabolism, we speculated that the location of lipid droplet, which is being hydrolyzed, that might affect, is it close to endoplasmic reticulum or is it close to mitochondria? And that may have because most of these channeling are stochastic in nature. So just the proximity could have a big role to play in this thing. So that's one big possibility. I agree.
Excellent. Thank you so much. I'm going to try to get started with my presentation, and we're a little bit behind schedule, but I'm hoping that I can catch us up. And I don't have to do too much background because Anand covered that very nicely. But so I'm going to be talking about triglyceride cycling and its role in energy expenditure in white adipocytes. And the point that I wanted to make here is that when we think about a white adipocyte versus a brown adipocyte and their roles in energy expenditure, we often think that the brown and the beige adipocytes with their higher mitochondrial content and UCP1 expression are really the dominating force in energy expenditure. But in the context of, trying to think about energy expenditure and obesity, there's just such a large mass of white adipose tissue that I think it's worth taking a little bit of a closer look at what's happening in these cells. So I think everyone here probably is familiar with this basic metabolism of white adipocytes, where glucose and fatty acids are taken up from the circulation and converted into triglycerides for storage in the lipid droplet. And when we want to access the stored energy in the adipocyte, we mobilize the triglyceride stores through lipolysis. And this is a process by which the, am I just gonna laser pointer? The triglycerides are sequentially, the fatty acids are sequentially cleaved off of the glycerol backbone, resulting in the release of free fatty acids and glycerol. Now, of course, we can also cycle these fatty acids, as was the topic of the previous talk. In order to do that, we need to activate the fatty acids into fatty acyl-CoA. This does take 1 ATP, but it's converted into AMP, so it's an equivalent of 2 ATPs to ADP. And then you also need to activate the glycerol. So this is how we get to a calculation of 7 ATP per cycle. And that's one of, quite a shocking number, whereas a lot of other cycles require only 1 ATP. And when this is happening in the lipolytic adipocyte, we see a big increase in oxidative metabolism. So I just want to show a picture. These are multilocular cells that we're using in these experiments. We try to push them to have larger lipid droplets. These are the the stromovascular cells of the inguinal white adipose tissue from young mice, which we proliferate 1 or 2 passages and then differentiate into adipocytes. And we use these for our experiments because we see that they have really robust rates of lipolysis and active metabolism. And so when we take these cells and do a seahorse experiment to measure oxygen consumption, if we activate lipolysis, here we're using the CL243, no, I'm going to trip over the numbers. The beta-3 adrenergic receptor agonist, CL compound, to activate lipolysis in the mature adipocytes. We see, activation of lipolysis is very rapid. We're activating this around 18 minutes. And then within just, 5 to 10 minutes, we see this robust increase in the oxygen consumption rate of these cells. And we know that this is related to the activation of lipolysis, because if we use that ATGL inhibitor, so you'll remember from the previous talk, this is the first enzyme involved in the cleavage of the first fatty acid side chain off of the triglyceride. So the inhibition of ATGL blocks lipolysis without blocking the upstream pKa and catecholamine signaling, and we lose this induction of oxygen consumption rate. So, one of the first questions is this simply the triglyceride cycling that we're observing? And in order to answer this question, we did some experiments where we used triacin C, which is an inhibitor of the acyl-CoA synthase, and it inhibits the activation of the long chain fatty acids into the fatty acyl-CoA. So here, if we look at ATP levels, and when we activate lipolysis, we see a reduction in ATP levels. And because we know from years and years of experiments, the main ATP demand in a lipolytic adipocyte is the demand for triglyceride synthesis. We assumed that if we, why can't I go forward? apologies, there we go. If we blocked the activation of fatty acids and therefore the utilization of ATP for the activation of fatty acids using triacetin C, that we would protect the ATP levels. And in fact, what we saw is even just the addition of the triacetin C itself without the activation of lipolysis actually had a surprisingly negative effect on cellular ATP levels. And in the context of lipolysis, there may be a trend towards slight protection of the ATP levels, but it is not significant. We still see that those ATP levels come down significantly from baseline. So this suggests that something else is affecting the metabolism of these cells. And when we look at oxygen consumption rate, we see that there's this disconnect between the ATP utilization for triglyceride cycling and the induction of the oxidative metabolism. So here we added the triacetin C first, and even just with the triacetin C, we're seeing a little bit of an increase in oxygen consumption rate. With time, that goes up more. And if we look at the effect of then activating lipolysis, so the normal lipolysis is here in pink, and the rate of oxygen consumption when we pre-inhibit with the triacetin C is here in the purple. You see that it's even higher. So this is really inconsistent with this increase in oxygen consumption rate being driven by ATP demand for lipid cycling. And this is going to come back later in my talk. I'm going to try to go relatively quickly, but I just want to mention previous work that we've done looked at how reesterification is regulated in white adipocytes that are actively engaging in lipolysis. And what we found is that the increase in intracellular fatty acids with lipolysis results in a serine phosphorylation of the STAT3 protein, specifically at the lipid droplet. And this has a functional effect to inhibit the activity of GPAT3, which is the first enzyme involved in coupling fatty acyl-CoA with the glycerol 3-phosphate and initiating the reesterification pathway. And that this inhibition of reesterification was really important for activating respiration. So here I'm just showing some data where if you look in the wild type mice, when we activate lipolysis, we get a very strong induction of oxygen consumption. But in the adipocyte specific STAT3 knockouts, this is again the in vitro differentiated adipocytes, they show a defect in their ability to mount this respiratory response to lipolysis. And this is not related to a difference in their ability to engage in lipolysis. They both are secreting the same amount of fatty acids. So to really understand what is happening here, we want to look at we wanted to look at whether or not this is coupled or uncoupled respiration. So in coupled respiration, we drive oxygen consumption due to ATP demand, but the effects that we saw of the triacetin C were inconsistent with our knowledge of ATP demand. When you uncouple respiration, uncoupling the electron transport chain from the generation of ATP, you will often get a dissipation of the membrane potential. And that's exactly what we see. And this, again, is an observation that goes back in the literature to the 1960s and 70s, that when you activate lipolysis, you see this rapid reduction in mitochondrial membrane potential. And again, this is related to the fatty acids, because if we prevent the fatty acid release, then we don't see this effect. So just to make sure everyone is on the same page, this is a general close-up of what's happening in the mitochondria when we are engaged in coupled respiration. We have reducing equivalents from the TCA cycle that are driving the electron transport chain and generating the proton motive force. This is then utilized to generate ATP. And importantly, this ATP generated in the matrix needs to get out into into the cytosol to be utilized throughout the cell. And this is done by the ATP-ADP carriers, which engage in a conformational shift to take matrix ATP, move it into the intermembrane space, and then at which point they can bind to ADP and move that back into the matrix. And with this and with the creatine cycle, we get the production of ATP and coupled respiration. And this is associated with a high membrane potential and a very high rate of ATP cycling by the ATP-ADP carriers. Now, when we think about the classic version of uncoupling, and actually there's still some lack of clarity in exactly how this works, but this is the model that I prefer, is that, you know, we know that the uncoupling protein 1 is dissipating that proton motive force and we lose the membrane potential. And UCP1 requires a fatty acid cofactor, and it seems that the fatty acids are acting as the protonophores that carry the proton across the membrane. And it's likely that the function of the uncoupling protein is really to facilitate the return of the anionic deprotonated fatty acid into the intermembrane space, as this is, you know, an anion, and the rate of spontaneous flipping of the anion is quite slow. So are we just looking at UCP1 mediated uncoupling in these adipocytes? And one thing that I wanted to look into is, you know, do we have beiging of these adipocytes? Sometimes you get UCP1 expression. Here we have a positive control for UCP1. This is a dark exposure. We just are loading 2 micrograms with a 10-fold dilution of protein from the brown adipose tissue. And then these are our adipocytes. And we even tried to do a 10-day stimulation with CL to promote beiging of these adipocytes. And we really were not able to detect any UCP1 in these cells. Whereas, and this is actually the mitochondrial fraction of the cells, whereas even in the whole cell lysate, we can very easily see the mitochondrial AAC2 protein, which is one of the most abundant mitochondrial proteins in these cells. Nevertheless, to really, you know, define whether or not this is UCP1 mediated, we did a UCP1 knockdown using a lentivirus with puromycin selection. And we saw that there was really no impact of the knockdown on the depolarization of the mitochondria. If anything, it went down even faster. But so this really indicates that it's not, you know, UCP1 is not present in these cells and does not seem to be promoting this uncoupling that we're observing. And so what this leaves us with is actually approach the role of the AACs in uncoupling. And this is another thing that's been known to occur for many, many years. And there was some really beautiful data that came out in 2019 by Ambre Bertholet that showed that the ATP-ADP carriers can also interact with a fatty acid and again facilitate the return of the anionic head into the intermembrane space, thereby creating a cycle that results in mitochondrial uncoupling. However, this pathway has been difficult to show in cellular systems because while UCP1 can utilize the fatty acid cofactor at relatively low physiological fatty acid levels, the level of free fatty acids required to activate this uncoupling by the AACs is really very, very high. And what I believe is happening is that the white adipocyte is a unique context in which we have when we activate lipolysis and the lipases are working at the surface of the lipid droplet, releasing those free fatty acids from the lipid droplet, and they flood into the cells and flow through the membranes, then they can get to very high levels under the physiological context of activation of lipolysis within the white adipocytes where we see this pathway activated. So I believe when we look at our cultured white adipocytes, we can see this quite clearly. So to test whether or not the ATP-ADP carriers are involved, we use pancreatic acid. This is an inhibitor of the ATP-ADP carriers. It locks them in the matrix-facing conformation so they can no longer cycle. And we see that, first of all, we lose our baseline coupled respiration when we inhibit the ATP-ADP carriers. But then when we activate lipolysis, we see a defect in their ability to respond to lipolysis with an increase in respiration, and it completely protects their mitochondrial membranes from depolarization. So is this simply a phenomenon of insufficient fatty acid buffering in our cell culture system? So we wanted to look at what happens in vivo. So first, we're doing these experiments in lean room temperature housed mice. These mice have very active brown adipose tissue. And one of the things that I'd like to point out is that UCP1 is the only thermogenic pathway that is independent of the AACs. Whether you're doing creatine cycling or other futile cycles that consume ATP, you need the activity of the AACs to provide to get the ATP into the intermembrane space in order for those cycles to go forward. So here we're doing an experiment in the lean mice. We anesthetize them with pentobarbital, so we eliminate the muscular movement and thermogenesis from that. So you can see even at baseline, after the injection of pentobarbital, their core body temperature goes down due to the lack of movement. And we pre-treat them with the pancreatic acid, and then we activate lipolysis. And what we see is that when we activate lipolysis, we see this nice increase in core body temperature, which is accompanied by an increase in oxygen consumption and carbon dioxide production. And if we inhibit the ATP-ADP carriers, we see a slight attenuation of the thermogenic response to lipolysis, but it's largely intact in these mice. And this is consistent with the current understanding that thermogenesis in these animals is primarily mediated by UCP1 and brown adipose tissue activity. However, when we look at the obese mice that have been housed at thermoneutrality that have low baseline brown adipose tissue activity due to the thermoneutral housing and very large amounts of white adipose tissue due to the diet-induced obesity, we now see that we still can have a very nice response to thermogenic response to lipolysis, but now when we inhibit it with the pancreatic acid, we really lose that effect. So meaning that these animals are dependent on AAC for this thermogenic effect and not UCP1. So in order to try to connect that back to, is this ATP futile cycle, or is this really ACP mediated AAC mediated uncoupling, we went back to our mouse model, which is the adipocyte specific STAT3 knockout mice, which have a defect in their thermogenic response to lipolysis and cold sensitivity. So in these mice, there's no defect in lipolysis, but under conditions where lipolysis is active, they have a defect in the adduction of energy expenditure. So here we're just showing that the high-fat diet mice, the knockouts have a decreased cold tolerance, and if we take them from thermoneutrality and acutely expose them to cold, then we see that they have a defect in their ability to maintain energy expenditure in the context of cold. Now, importantly, this is not a brown fat defect, because when we look at this knockout in the normal diet mice where brown fat is dominating, we see no defect. So what we decided to, so then we also did the pentobarbital experiment in these animals. And what we see is that while the wild type animals can increase their core body temperature in response to the activation of lipolysis, the adipocyte specific STAT3 knockouts are really defective in that response. And this is despite the fact that their serum free fatty acid levels are indistinguishable between the two groups. Further supporting that this is not a brown fat phenotype, we do not see this effect when we look at the obese UCP1 driven brown adipose tissue STAT3 knockouts. And finally, what we see is that we can actually recover this defect simply by restoring the inhibition of reesterification. So here we used an inhibitor of DGATs before we activated lipolysis, and then we were able to recover the thermogenic response in the knockout. So the knockout, everyone got CL in this experiment, and so the knockout with CL and no DGAT inhibitor has a defect in the response. But when we provide the DGAT inhibitor prior to the activation of lipolysis, we see that we can recover their ability to mount this thermogenic response to lipolysis. So we're getting very close to the end of our time, so I'm going to finish up. I think that this is really an interesting phenomenon, and I would never argue that a white adipocyte is better at energy expenditure than a brown adipocyte. We know that brown adipocytes just have so much more mitochondria and dense cristae packing. But when we have a huge amount of white adipose tissue, this may actually be an avenue for trying to promote energy expenditure by impacting fatty acid deposition within the white adipocytes. And I would like to thank all of the people who contributed to this work. I tried to put the pictures throughout my talk. Melissa and Zane are technicians in my lab right now who have been working very, very hard, and some former lab members, including a rotation student who really did a lot of those BKA experiments. And finally, Miriam Ahmadian, who has really contributed to the field of lipolysis research. So thank you, and I will take questions with the few minutes we have left. Anand, do you want to get us started? And then hopefully we have some attending questions too.
Yeah. Hi, Shannon. Great talk. Yeah, I'm curious about the DGAT inhibitor in your knockout mouse. So how do you think it's working? Is it through the fatty acid channeling, or because we have seen that with the DGAT inhibitor, it does increase fatty acid oxidation. So do you think that's the mechanism, or that's one point? And second was in your schematics, I saw you show CKBs. And I'm wondering if you believe, or what is your opinion on the creatine cycle in white adipocytes, let's say, because I think Lawrence's data is mainly on brown and beige. So yeah.
Yeah, so what I think, let's see, for the effect of the DGAT inhibitor, what I think is happening is that the regulation of reesterification in the adipocyte is really important for as a way to modulate those fatty acid levels. So while on the one hand, the reesterification pathway is energetically expensive, it's more energetically expensive to have those free fatty acids. And it's important to note that the uncoupling is mediated by the free fatty acids and not the fatty acyl-CoAs. And those free fatty acids can diffuse into the mitochondrial inner membrane without the assistance of a transporter. So when you enhance esterification, you essentially have a sink for reesterifying and neutralizing those free fatty acids. And so that's why I think, but also there's like, kind of a teleological argument that when you're engaging in lipolysis, you may not want to reesterify, you may want to have some regulation of whether or not you're reesterifying. And here we get into like a really interesting situation where I think we have to take into account what's happening between adipocytes as well as from adipocytes to endothelial cells, and whether or not those fatty acids are reaching the circulatory system and what the feedback mechanisms are there, because I think that those are really interesting. And sorry, your second question was related to
CKB.
Oh,
Just
yeah.
your opinion, not a
So
question, but yeah, I don't know.
I don't think that in a bona fide white adipocyte, we're really seeing much creatine cycling. I mean, of course, we have the normal role of CKB in the export of ATP equivalents from the mitochondria. But I think the localization of the phosphatase in the white adipocytes does not suggest that that's going to be an active thermogenic pathway in white adipocytes.
Thanks. I agree. I mean, with the DGAT inhibitors, because in our previous paper in molecular metabolism, because when we add DGAT inhibitor, the oxygen consumption goes up, at least in vitro. But a lot of it was dependent on fatty acid oxidation. So we believe that because the DGATs use acyl-CoA, and then that is directly converted through CPT1 for oxidation, when it cannot be reesterified. But the phenotype, I think, is the same. So who knows the details? But yeah, it's exactly the same. Thank you.
Yeah, I think it's interesting to think about because that's another point of what happens, like whether or not the fatty acids is converted to the fatty acyl CoA, and then whether or not the CoA goes to oxidation or esterification.
Exactly.
Do we have any questions from the audience? I know we've already gone a minute over. Yeah, can we get Daniel unmuted for his question?
Shannon, I just have a real quick question. That was a great talk. But what you just said was that some of this is mediated by free fatty acids, not acyl-CoA esterified going into the mitochondria. Is that correct?
Correct.
So how does it get in if there's no need for CPT1?
Well, so this is what's I think is really interesting. Free fatty acids, because of their chemical nature, are really able to efficiently flip through membranes. This happens on the order of, when they're protonated, it's very fast. It's seconds. When they're deprotonated, then it takes a little bit longer, but it's more like minutes, 5 minutes, 10 minutes for you to get the cycling of the deprotonated fatty acid. So when we have these really high rates of release of free fatty acids from the lipid droplet, those fatty acids are going to be, some of them are going to bind to the fatty acid binding proteins and be transported. And if they go out of the adipocyte and reach the circulatory system, get bound to albumin, they're gone. But they can also go on and off of the binding proteins relatively rapidly, and the exchange from the binding proteins to the membranes is also quite rapid. So essentially the binding proteins are helping them to diffuse throughout the cell, but every time they're knocking into a membrane, they have a very rapid on-off ability to go into that membrane and to flip to the other side of that membrane.
Yes, thanks.
And we have a question from Lindsay.
Hello. Yes, I had a follow-on question from that then. So if fatty acids are able to flip across the mitochondrial outer membrane, they'll then get into this intermembrane space. Is that filled with fluid, I presume?
Yes, yes, because that's where you have the very high concentration of protons. And so that is going to help to protonate those fatty acids and to facilitate the transport then of those protonated fatty acids through the mitochondrial inner membrane. And where the ATP, ADP carriers will jump in is when the concentration of free fatty acids in the inner mitochondrial membrane. So you imagine if they go from the intermembrane leaflet side of the inner membrane where they're going to be protonated and they flip to the other side, they're going to lose that proton because the proton concentration on the other side is so much lower. Then the ATP, ADP carriers facilitate the return of the deprotonated fatty acid to the intermembrane side of the leaflet, where it can then pick up another proton and flip through again.
Okay, so what I was trying to kind of figure out is if obviously, so there's no need for these generally insoluble fatty acids to be solubilized by a lipid transport protein at any stage then. And, you know, once they've reached the sort of matrix, if you like, and inside, you know, in the heart of the mitochondria and also in that intermembrane space, or is that just not necessary?
So this is a really good question, and I think that this is where we need to really think about whether we're talking about the fatty acid in solution or the fatty acid in the membrane. So when they do have some very sparing solubility in solution, but you really need the fatty acid binding proteins to solubilize them in an aqueous solution. But the membrane is an entirely different chemical environment. So the membrane is really ideally suited for a free fatty acid. Their carboxyl heads can be very happily on the surface of the membrane. They have the polarity that's going to enable them to interact with the aqueous environment on the other side of the membrane. And then inside the membrane, where you have all of those acyl chains of the different phospholipids that make up the membrane, the acyl chain is quite happy and quite soluble in that environment.
Okay. Thank you.
Well, thank you.
There is
yeah.
a question in question and answer, Shannon. Maybe you can check from Denny Blondie.
Oh, okay, yes. Yes, okay, so this is a question that relates to in the BKA experiments, how can we distinguish whether the reduction in respiration is due to the impaired proton leak versus impaired ADP transport? And this is a really critical question to answer. So of course, when we inhibit with BKA, we block both coupled and uncoupled respiration. We see that in our baseline effects in the cell culture. And of course, in the mouse model, when we're treating with pancreatic acid, we really cannot differentiate between the effect of inhibiting the coupled respiration and the effect of inhibiting the uncoupled respiration via the ATP, ADP carriers. However, in the cell culture model, we have a lot more tools to be able to look into this. So we have a few pieces of evidence that really help to support this. So one thing is that when we do these experiments with oligomycin to block ATP synthesis, we see the same baseline reduction in, let me just try to go back to it because I have, this one, share. And then, oh, here, yes. So with the oligomycin, again, we see this loss of baseline coupled respiration. But now when we activate lipolysis with the oligomycin, we see that the respiratory rate increases very rapidly, and they have no defect in their ability to mount this uncoupled respiration that's associated with loss of membrane potential. So it's really the difference between and the effect of the BKA in the context of oligomycin that we're able to differentiate the effect of the uncoupling versus the effect, their critical role in replenishing the ADP stores to allow for ATP synthesis. Yeah, so I think hopefully that answers the question. And I will stop sharing again. And I think that, oh, many of you have held on. Thank you to all of you who have stayed to this point. I'm happy to stay and answer any more questions from the group. No, thank you, everyone. I appreciate everyone being here today, and I hope everyone has a wonderful afternoon. Thank you, Anand.